So a few things. One is, let's start by looking at this warm up. That's what we kind of been doing all the time, is kind of doing the warm up and checking it out. So I'm going to bring up. Take me just a moment. Human anatomy. All right, so I'm going to share my screen and we can go into modules, our warm up for today, and we can just take a preview and take a look. And does 15 minutes, does that feel, I figure that makes you feel at least you've got a little more of a, because normally it's a quiz, the clock's ticking. So I put 15 minutes. It's definitely more time than you need, but at least to kind of give it a sense that you're supposed to kind of do it in a limited amount of time. So question one, part of the brain involved with endocrine control, hunger, thirst, emotions, body temperature, among other things, would be what?
Hypothalamus.
And that's totally going to be the hypothalamus. So, substantia nigra, that is, where do we find the substantia nigra?
In medium brain. Midbrain.
Yeah, that's going to be in the midbrain. That was the part that was projecting up towards the basal nuclei. That's the part of the brain that starts degenerating in Parkinson's disease. So it's not substantia nigra. This I've mentioned before. What is decussation of the pyramids?
Decussation, it's a crossover.
Yeah, so decussation is that crossing over from right to left. Pyramids are the axonal tracts coming from the primary motor cortex. So this is the kind of place along the brainstem where the axons that are going to your skeletal muscles are crossing over from right to left, left to right. Where would you find the pineal gland?
In the epithalamus?
Yeah, that's in the epithalamus. That's up there in the roof of the third ventricle there. And it's making melatonin. Medulla, so the medulla is involved with a variety of autonomic functioning like cardiovascular and respiratory, but it's not doing these drives like hunger and thirst. It's not doing emotions. It's not doing endocrine control in this way. Hippocampus was the part that is in the floor of the temporal lobe that was doing the, as part of emotions, also really important for what we call consolidation of memories. We're not going to go so much into that, but in other classes in physio, we talk about the hippocampus as essential for converting your short-term memories into long-term memories. Corpora quadrigemini, this was the superior inferior colliculi, which are involved with what? What do these guys do? And these are also midbrain.
Those are visual and auditory reflexes.
Yeah, visual and auditory reflexes, kind of, whoa, what's that? Or, ooh, don't want to run into that. So kind of these more kind of low level kind of reflexes with odd hearing and vision. So hypothalamus, kind of a long way of saying, yeah, you're right. Hypothalamus, this is the thing that's doing all this stuff. All right, continuing. Oops, wait a second. I'm in this. I want to stop my back.
You also said something about saccades, so that has to do with the visual.
The superior colliculi in the corpora quadrigemini are the things that calculate the saccades. What what is the motor program you have to run with those extrinsic eye muscles, superior, inferior, medial, lateral, rectus muscles? What do you have to do to move the eyeball just right? So, first I'm staring at the picture of Jamba, and now I'm staring at the picture of Bella. So that is a saccade. You're staring at one thing, and then all of a sudden you're staring at something else. And that is the neurons that do that motor programming is in the superior colliculi. All right. Oh, mouse. Okay, now I'm back to my mouse. So now, oh shit, all that stuff I drew on there is still on there. Let me clear the drawing. All right, I'm still learning this. All right, here we go. What area of this is also, I really wanted to figure out a way to just have question 2A and 2B, and it will not let you. You have to, so this is my best way to do it. So what is this part of the spinal cord right there?
Ventral horn.
Yeah, this is the ventral horn. And how do we know it's the ventral horn?
It's rounded.
Rounded.
Yeah, so it's more rounded. It's kind of harder to see here. Typically, the ventral horn is going to also have kind of a deeper fissure here, and the dorsal side has that sulcus. It's more of a rounded. In fact, I don't know if I've mentioned, one of the ways I used to remember that you have kind of a deeper fissure on this side is like ventral or anterior, which are both synonyms. Letters look kind of more peaky, whereas dorsal or posterior is kind of more of a shallow, round kind of look. So when I was trying to remember, like, wait, which one was deeper? Which one was more kind of a shallow sulcus? That was kind of how I would remember it. Like, and then I would also, but then when you're talking about the horns, the posterior is more peaky looking. So, whatever kind of mnemonic you come up with. So this is going to be the ventral horns, and what happens in the ventral horns?
All the motor activities.
Yeah, that's going to be all the motor activities.
For that question, I also added a word, ventral root horn. Will that?
So I'd give you partial credit. The root is something a little different. The root is the neurons. Let me get my drawing here. The root is the axons that are coming out from here. So we have.
Yeah, I realized that later. And then I'm just concerned about the score that you're going to give me for that, because I added the word root and horn, ventral root horn.
So what in fact, maybe I'll write this out specifically so it's, because I can say it, but then it's probably better if I just write it. Whiteboard. So in taking warm-ups, if the automatic grading is off, I mean, by off, I mean doing something wrong. I should say incorrect. Just let me know. So send me an email. And I will fix things. Because the reality is, when you're making these quizzes, you have to, the program is really like rigid about what it assumes is correct or incorrect. This idea of partial credit does not make any sense. You're either you're either with me or against me as a student. You can't like kind of know it, but not it's not quite right. The program is either you're right or you're wrong. But obviously, in reality, that's not the way life is. That's not the way I like to grade. So if you think either you got it right and the program messed up, or maybe it's not totally right, but it should have partial credit, just let me know and I will go in and I will kind of adjust things. So that is that. And now back to our regularly scheduled program. So question 4, which is actually question 3, and again, I have not figured out how the heck to, I don't think there is a way. I actually called it question 3 and question 2A, but it doesn't show up here. So this is match the muscle with the nerve. Biceps brachii. What is the name of the nerve that goes and will innervate biceps brachii?
Cutaneous.
Musculocutaneous nerve.
Yeah, it's going to be musculocutaneous. That's that one that goes, and it's also going to do the other flexors of your upper arm, like the like brachialis muscle. So if I go here, it would be musculocutaneous nerve. Flexor carpi ulnaris. That's the one. Remember the like FCU? It's the one that wants its own nerve.
Ulnar nerve.
It's that ulnar nerve. And the ulnar nerve, which is also taking like feeling from kind of the pinky side of your hand. That's why like when you dig in on the ulnar nerve where your funny bone is there, you can feel tingling on your pinky side. Vastus lateralis, which is one of the quadriceps muscles. What's the nerve that goes to the front of your thigh?
Femoral nerve.
That's the femoral nerve. What plexus is the femoral nerve coming from?
The cervix.
Lumbar. Lumbar.
So so this is where it's going to be actually helpful to remember. It's the femoral nerve comes from the lumbar. So a little higher up on the spinal cord. Whereas when you get like the sciatic nerve, which is going to go down the back of your thigh, is starting from the sacral region. So when you're looking at a spinal cord or a picture or a model, and you're trying to like make sure, like, wait, is that the femoral nerve or is that the sacral, the sciatic nerve? Particularly because these pictures and models tend to be tricky trying to figure out like depth. Like one of the things I've been really realizing looking at like pictures of models is you really totally lose the sense of depth. Like what's in front, what's below, when you look at something for real, you get a sense it's got this three-dimensional quality. You'd hold it and but when you look at a picture, everything is just flat. So in particular, you're going to want to make sure you keep track of femoral nerve is coming from the lumbar plexus. What was the other nerve we talked about that comes from the lumbar plexus?
Obturator.
The obturator. Except the obturator, instead of coming down the front of the thigh, is going to go down through that obturator foramen to go to the inner thigh. Triceps brachii. What's the nerve for that one?
Radial.
That's the radial. So the radial nerve is basically doing all the muscles on the back, all the extensors. It's doing triceps brachii. It's doing all the extensor muscles, extensor carpi ulnaris, extensor carpi radialis brevis and longus, extensor digitorum. So all of those. And like I said, the other thing that does not match is it also does brachioradialis, which is a flexor, but it does that as well. So that was question 3, which here it says is question 4. So question 4A. Oh, let me clear my little pictures again here. Clear drawings. So this nerve here, to figure this out, so which plexus is this arising from? That's, I guess, the first thing we should say. What's the plexus?
Brachial plexus.
Exactly. It's coming from the brachial plexus. And when you're trying to figure out the brachial plexus, you kind of come up here. I tend to like look for that M, that or W or whatever you want to call it. So like right around here, here's where you're going to have the different chords. Again, this is where this is going to be the lateral chord. Well, actually, this is the lateral chord. This is the medial chord. On this one, it gets really tricky. The posterior chord is totally not clear that it's actually behind it all. This chord right here actually is going behind, right? So that this would be creating the radial nerve. But what you're going to be noticing is this M, which has got to be the lateral and medial chord, and then goes to the median nerve, which is the one that arises from the middle of it. And so this is going to have to be the median nerve. Clear drawings. Actually, get my mouse back. And it says, name a muscle that's innervated by that nerve.
Palmaris longus.
Yeah, so palmaris longus would work, as well as flexor carpi radialis, as well as the medial portion, the lateral portion of extensor digitorum superficialis. So that is that. And I'll finish this. We're going to come back to things that look like this in just a moment. Question 7. Collection of nerves that extend below the end of the spinal cord to exit at vertebral levels below L1 is called?
Cauda equina.
The cauda equina, again, which means
Horse's tail.
Horse's tail. Because basically you've got the spinal cord comes down. What do we call the very end of the spinal cord?
Filum terminalis. No.
Nope.
The conus
Something. Corpora.
The conus
Phylum.
Yeah, that's the conus medullaris.
Medullaris.
You can probably tell I am writing with my mouse. Conus medullaris. Then what do we call the little extension of pia mater that anchors this down at the base of the spine?
phylum
Phylum
terminal.
So that's the phylum terminale, is this little extension of pia coming down. Where would I find the Crista galli?
Crystal galli on the sphenoid.
Yeah.
Ethmoid bone.
Oh, not on the sphenoid. You're close.
Ethmoid bone.
It's on the ethmoid bone.
Oh.
It's on the ethmoid bone, but it's also named after an animal. Cauda equina means the horse's tail. Crista galli means the coxcomb. What is the name of the dural septa, the little sheet of dura mater that anchors on the Crista galli in the skull?
That's called the falx cerebri.
Yeah, the falx cerebri. So yeah, make sure, as again, remember, you always want to review things. Like, as we keep going and learning new stuff, it's kind of adding to a bigger picture. So cauda equina is going to be all of the nerves that leave the spinal cord, but then are going to leave levels, at vertebrae below L2. Conus medullaris is around L1 or L2, but you have spinal nerves leaving at lower lumbar and sacral regions. So all of the nerves that are coming down end up looking like a horsetail. So horsetail. Continuing down. Oh, I think that's it.
Did you
Um,
say the sella turcica is on an ethmoid bone?
Sella turcica is on the sphenoid. The
Sphenoid
crista
bone.
galli is on the ethmoid bone.
Yeah. Okay.
Clear drawings. So one of the things that I did was I took a picture. We have a model. There's this big model, a SAMSO model, which is, in fact, let me go into our account. Doobie-doo. If you go into modules, and then, oh my God, there's a new module here now for us called Anatomy Lab Model Pictures. And there's NerveMan, SAMSO NerveMan. So this is actually this really big thing. It's like about 4 feet tall. It's about half real size. I actually had to like stitch together 2 pictures to make the picture. So it looks like this. And there's 2 things we have. One is there's a key. So this is the key that has like the different numbers and which nerve or whatever that it goes to. But then there's a big JPEG of the actual model itself. So here it looks, I actually did it at a high enough resolution so you can actually kind of move around and actually, so here, when you're looking at the brachial plexus, here are the nerves and the trunks. And, if we do our annotating again, draw, again, this is going to be, maybe let's do it in red, might be easier to see. We're coming down again. Here we find the M is here, which means this has to be my lateral cord. Here's the M. So this side, this has to be my medial cord. Which means if I'm continuing along the lateral cord, what nerve am I looking at here?
Musculocutaneous.
Exactly, musculocutaneous. If I'm following the medial cord going this way, what nerve does this have to be if I'm just staying medial?
Ulnar nerve.
That's the ulnar nerve, exactly. And then if we take the M and we look at this muscle that's formed by contributions of both the lateral and medial cord, then this is going to be the median nerve, which we saw on our quiz. And here you get a little better sense of this other cord here. This is the posterior cord. And here you can kind of see, oh, it's kind of going underneath. Kind of staying underneath, going underneath. So this is actually going to be the radial nerve here. But you can see that it's actually a little tricky to see because you have to like, if this was actually three-dimensional, something that you could like poke at or lift up, you would definitely see, oh, this is going behind. But you have to, it's a little more ambiguous. This posterior cord, what's the other thing? This is going underneath as well and innervating around the shoulder here. What going under the arm?
Axillary.
That's the axillary. So this is kind of giving you a sense of like on a lab test that we will hopefully have if we go back to school. You know, some of this could be stickers on like the nerve man. So you should be able to look, oh, whoopsie daisy. You know, sometimes it gets trickier over here because the arm is not in anatomical position. You know, here we have the median nerve, but then we have, if we look at the posterior cord, you know, here's the like radial nerve, but it's a little trickier to see. This is the posterior cord going behind. So just kind of letting you know because things are twisted around. We also have other drawings and pictures. Sometimes that's why I said it's like important to really learn the brachial plexus, because once you're trying to figure out which one is which of these, even though I'm not going to test you on details of the plexus, unless you know how to navigate this crazy mess here, you're not going to know what nerve you are at when you actually get to these nerves in the arm. Mouse. When we get, yes, let me clear my drawings again. Here's where I stitched together the picture so it's not beautiful. Again, I was just doing this quick. Maybe I'll have time to make a better picture version of this. But the basic idea here, like when you're thinking about the different plexuses, you definitely want to realize, oh, this would be kind of lumbar region. This is going to be more sacral region, like sacral versus lumbar. So when I'm thinking about the different nerves, if I have a nerve, well, let me get my mouse back. Oh, you fucker. Oh, sorry, excuse me. So if I'm going to get back and like kind of highlight, you know, this is kind of the more lumbar region. This is more the sacral region. And then I can look at the nerves that are coming down. This nerve here, which is kind of, you can see going in front of the hip bones, and you follow and you can see it's coming all the way up to the lumbar. So I know that that is going to be which nerve?
Femoral.
That's the femoral, exactly. This one here is a little trickier to see because it kind of what's in front, what's in back, but this is also, what nerve is this one here?
Obturator.
That's
Or
the obturator. Yeah, exactly. That's the obturator. It's kind of going through the obturator foramen, and which you have to just pretend is there. But then you can also see it's starting from more of the lumbar region. Whereas if we look at this big honking nerve here, which one is this probably?
Sciatic.
This is going to be the sciatic. It's starting from the sacral region and it's kind of going down behind on the back part of the hip. And this one does not have, the pudendal nerve would kind of be, just kind of go like this, coming out of the sacral region and just kind of go right there. But so when you're trying to recognize the femoral versus sciatic, you definitely want to keep track of whether or not it's starting from the lumbar or the sacral region. That's going to be an important clue so you don't get tricked, particularly in these drawings where sometimes it's kind of hard. Everything's lying on top of each other, and it's the more clues you have, the better chance you're going to get it. Um, so that is that. Any questions about the stuff that we went over last class? All right, so what we're gonna do now is we are gonna continue on into the peripheral nervous system. And let's see, let me get back to my little stylus here. Booby-doo. Okay, dokie. So far we've been just talking about the different nerves that are carrying the axons in and out of the to the central nervous system. On those nerves, you can have motor, you can have sensory, you can have autonomic, you can have somatic. So we're going to do now. So we've just been kind of talking about the cabling. Now we're going to go more into the different functional parts of the nervous system again. So let's start with somatic motor. So again, what is the somatic motor doing? What does it control? This is a division of the PNS. What's up, buddy?
It's skeletal muscle and voluntary movements.
Exactly. So this is the one that does voluntary muscles. Basically, this the skeletal muscle. Excellent. And the way it's hooked up is pretty straightforward. So that's why we usually start with this, because it's pretty straightforward. So we're going to have the brain and the spinal cord, aka the CNS. And the target for this, the thing that this is going to control, is going to be some skeletal muscle. The signals, in fact, what part of the brain do the signals start in? When you're controlling your motor stuff, we talked about the particular part.
Precentral gyrus.
Exactly, the precentral gyrus. You're going to have, but eventually you're going to have signals coming down the spinal cord. In general, there's going to be some neuron. It's going to have its cell body here in the CNS. And then we'll have an axon that goes all the way to the muscle. There's going to be one neuron with its soma, its cell body in the CNS, either the brain or spinal cord, an axon that goes all the way to the target muscle. Muscle. Now that means that you have these really long neurons, right? You've got some neuron that goes from your spinal cord all the way to the tip of your toes for the little muscles that you're using to wiggle your toes around. Those things might be like over 3 feet long. So this is a really big, long neuron, or it can be. We've spent some time looking at the neuromuscular junction when we were doing the muscle system. What is released by this somatic motor neuron here at the muscle?
Acetylcholine.
Exactly. Somatic motor neuron releases acetylcholine. And then the other thing we can put in here is that the skeletal muscle has these receptors. There's a subtype of acetylcholine receptors called nicotinic cholinergic receptors. So muscle, in fact, I'm going to do this using my little keypad because it's not going to be as messy looking. So I'm going to just type this in. I'm going to say text. So, well, that's too small, isn't it? Muscle has what are called nicotinic cholinergic receptors. Nicotinic cholinergic receptors are always excitatory. What that means is if you send a message, an action potential down this motor neuron, you are going to cause the skeletal muscle to contract. We're going to see this is going to be different when we get into the autonomic motor. When we were looking at autonomic motor neurons, sometimes they release acetylcholine and they might actually relax the target muscle. And that's going to be because there's a different kind of receptor that can have more variation in response. These nicotinic cholinergic, they get their name nicotinic because they bind nicotine. That's how come they get their name, is they actually bind nicotine. Why does nicotine actually affect your, it's kind of a stimulant, helps you focus? It's because it's binding these acetylcholine receptors in your brain. These aren't just on your muscles, they're all over the place. So this is somatic motor. There's really not much more to say about it. It's pretty straightforward. For voluntary somatic motor, you have one neuron. Its body is in the CNS, and then the axon takes you all the way to the target muscle. At the target muscle, you release acetylcholine. And if, and the muscle that's at the target has these receptors that are always going to be excitatory. So, ta-da, we've done that. Remember we talked about Botox? What does Botox do in this whole system here?
It blocks the release of acetylcholine.
Exactly. Botox blocks the release of acetylcholine, which means, since even if you're sending a message down here, if you don't release acetylcholine, then the muscle is never excited and it just stays kind of relaxed. And the muscle tension is kind of what gives you the crow's feet. So people, again, inject that around the eyes. So the muscles just relax, things look smoother, but again, lose the ability to convey any kind of emotional subtlety because they're all paralyzed. So any questions about somatic motor? All right, so now next what we're going to do is we're going to go to autonomic motor, which is much more complicated. So let's do it. Yep. So autonomic motor, the sympathetic and parasympathetic. Um, ack. So first I'm going to talk about the way these are similar. So things that are similar just as long as your autonomic motor. And then we're going to talk a bunch about the differences as well between sympathetic versus parasympathetic. So the way they're similar, and we're going to have the brain and the spinal cord, CNS. What is the target organ going to be for the autonomic motor system?
Smooth muscle and cardiac muscle.
Exactly. It's going to be smooth muscle, the involuntary, cardiac muscle, maybe speeding up or slowing down your heart. What's one other thing that it might innervate?
Glands.
Exactly, glands. This is going to be smooth muscle, cardiac muscle, glands. And so let's see, let's put like the heart here as our target. There's always going to be two neurons before you get to the target. There's going to be neuron number one that has a cell body in the CNS that comes out partway. Then maybe I'll do another one. There's going to be a second one, neuron number two, which then takes us to the target. What do we call cell bodies that are outside of the CNS, or that are in the PNS, I should say?
Ganglia.
Exactly, ganglion. So this is a ganglion right here. So there's always going to be a ganglion. And we're going to see it's if we're sympathetic versus parasympathetic, where the ganglia are and how they kind of show up is going to be different. But there's always going to be neuron one going to the ganglion. This is always going to just be a little, it's going to be an excitatory synapse here. Yeah. You don't have, you're not responsible for the details, but it's basically the exact same thing we saw at the skeletal muscle. This neuron one is releasing acetylcholine. The next neuron has nicotinic cholinergic receptors, which are always excitatory. So this is always just going to be kind of like a relay. You send the message from here to here, and then it continues on to go where it's going. What's going to be interesting is when we look at the actual target. That's where you're going to have differences and where things are going to be kind of fun. And so this is how these are both similar. These are both similar in that there are two neurons, neuron one, which goes to a ganglion, and then neuron two, which continues to the target. They're both similar in that they operate smooth muscle, cardiac muscle, glands. So now what we're going to do is look at how these are different from each other. So, and I like to always kind of try to do color coding. All right, sympathetic versus parasympathetic. I just did not color code it, did I? Let's do the blue. Parasympathetic. All right. So again, let me draw the brain and the spinal cord, so the CNS. I'm going to have our target, both cases, it'll be the heart. One difference in between these two is the length of the neurons for the first versus the second. Sympathetic is always going to have a short first neuron. We also call preganglionic neuron. And a long second neuron, which we also call the postganglionic neuron. So there'll be this first neuron that comes out, but very close to the spinal cord. Then a second neuron that then goes woo, all the way to the target. So the ganglia are near the spinal cord. I could just say, near the spine even. Parasympathetic. You have some initial neuron, and it goes a really long way, and then the ganglia is right near the target. So this is going to have a really long first neuron, or preganglionic neuron, and a very short second neuron. And the ganglia are, sometimes they call them terminal ganglia. Terminal, end of the line, because the ganglia is right near almost where the target is. Sometimes they're called intramural ganglia. Intramural means within the wall. Sometimes the ganglia are even within the wall of the target organ. So that's one difference, is sympathetic, short first one, ganglia near the spine, and then a long second neuron, whereas parasympathetic, long first one, and then a short little one to get the rest of the way. Another is where the first neuron arises from. For the sympathetic, we talk about the thoracolumbar outflow. So all of the first neurons start in the kind of either the thoracic or the lumbar region of the spinal cord. And then there's going to be parasympathetic, on the other hand, we talk about the craniosacral outflow. Meaning you either start with cranial nerves or you have sacral nerves. So, and again, the really long ones that go out someplace. So craniosacral outflow, meaning that the first neuron starts either as a cranial nerve or as a sacral nerve, whereas sympathetic thoracolumbar outflows comes out either the thoracic or the lumbar regions of the spinal cord. not only does parasympathetic originate from cranial nerves, about 70% comes from one particular cranial nerve. In fact, what is the only cranial nerve that actually leaves the head to go all over the place?
The vagus.
Exactly. So, cranial nerve number,
Number 10.
Number 10.
number 10. So, the vagus nerve does lots of things. It also does motor stuff. But I mean, it also does sensory stuff. But one of its important things is it is about 70% of the parasympathetic innervation of your body. Like it's like your heart. We're going to talk about this more later, but your heart has a natural rhythm of about, like if you just ripped somebody's heart out of their chest, it would start beating in your hand at about 100 beats per minute. But obviously, most people, their resting heart rate is around 70 or lower. And that's because you have constant parasympathetic innervation, keeping it a little slower. If you cut the vagus nerve, somebody's heart rate will speed up and go back up to about like that 100. So you've constantly got the vagus nerve talking to your heart, telling it, go a little slower. So vagus nerve. So sympathetic. So that's two of the things. And in fact, at this point, I am going to show you a, what do you call it? A PowerPoint thing. So I'm going to go to my remote instruction anatomy, Pearson, Mareb, autonomic. Okay, here we go. If we go to share screen, yes. Boom. All right, so yeah, just kind of remember peripheral nervous system, AC. If we go into, where's the pointer options highlighter? Peripheral nervous system is what we're talking about. Motor is what we're talking about. We've covered somatic motor, and now we're in the autonomic motor. So this is where we are right now. For somatic motor, there's basically one neuron that goes all the way from the CNS to the target muscle. It's simple as pie here, one neuron. For the sympathetic and parasympathetic, sympathetic is starting here in the thoracic and lumbar regions. There's the first neuron that goes to this ganglion. A second neuron then continues to the organ. For parasympathetic, the first neuron originates in the cranial or the sacral regions. The first neuron goes almost all the way to the target organ, and then the second neuron is often even within the wall of the target organ. We're going to talk about, there's one other thing that can happen with sympathetic that makes it a little unique in that, in addition to this, it can also target the adrenal gland to release adrenaline. But we'll talk about that in more detail in a few moments. Again, somatic motor, one neuron all the way from the CNS to the target muscle. Whereas here we've got, like this picture here is also kind of showing you this green. Is this green sympathetic or parasympathetic?
Sympathetic,
And how do we know?
because it's coming from the thoracic lumbar region.
Exactly. It's coming from the thoracic lumbar region, plus we can see the ganglia is close near the spine. whereas this side is coming from this cranial and the sacral region, and the first neuron goes almost all the way to the target. And then there's an itsy bitsy second neuron to finish the rest of the path.
Black.
Again, this is kind of just showing, long first neuron and then itsy bitsy second neurons for the parasympathetic. We're going to talk about this kind of stuff a little bit later in a few moments. So we're going to talk all about the sympathetic trunk in just a few moments. We're going to talk about this in a few moments. So let's get back to our whiteboard. Okay, so there's going to be one other big difference between sympathetic and parasympathetic that we need to talk about before we get into the details of some of this wiring. Sympathetic versus parasympathetic. The other big difference, and the one that comes up and being very important when we look at the pharmacology, like different drugs we have that are designed to either activate or suppress sympathetic or parasympathetic responses. What's nice is they tend to release different neurotransmitters at their targets. So in general, sympathetic is going to release either we call NE, norepinephrine, at your target, or sometimes epi, which is epinephrine. And this actually tends to be released just into the bloodstream. Epinephrine is the word we use in the United States. Other places they call it adrenaline. Epinephrine and adrenaline are synonyms. So an EpiPen, you're jabbing them, you're giving them epinephrine if they're in anaphylactic shock, basically brings their blood pressure back up and all that. It's adrenaline. You're giving them a shot of adrenaline. Norepinephrine is the one it's very similar, but it tends to be used as a neurotransmitter released in a synapse from one axon to a dendrite or to some kind of target. I shouldn't say dendrite, but some kind of target muscle. So the receptors we call adrenergic receptors. Adrenergic, because they bind basically adrenaline and noradrenaline. And there's different kinds. There's alpha 1, alpha 2 adrenergic beta, 1 adrenergic beta 2, etc. We're not going to get into the details here, but it's worth knowing that beta adrenergic receptor is just a receptor for epinephrine and norepinephrine. Because like beta blockers, if somebody takes a drug that's a beta blocker, that's a common thing you'd give them if they have high blood pressure. And it's basically blocking receptors on the heart that would normally like speed up your heart. So if you're trying to deal with keeping blood pressure down, you don't want the sympathetic nervous system to speed up the heart and kind of create increases in the blood pressure. So you give them beta blockers, drugs that block the receptors for adrenaline and noradrenaline on the heart. You can also have adrenergic agonists. drugs that bind and actually stimulate the adrenergic receptors, like the little asthma inhalers are adrenergic agonists, like albuterol. If you have an asthma attack, you take in your little inhaler, it's giving you a drug that binds these receptors and turns on the sympathetic response. And part of turning on sympathetic response is dilating your bronchial passages so you can breathe easier. So because sympathetic tends to use epinephrine and norepinephrine, you can use drugs that will preferentially block or activate the receptors for those in order to turn on or off sympathetic response. There are a few exceptions, like sweat glands are controlled by sympathetic, and yet they actually have acetylcholine in their synapses. So that's why I said in general, it releases norepinephrine and epinephrine, but not always. But for all intents, for our purposes, you can think of sympathetic as primarily epinephrine, norepinephrine. Parasympathetic, on the other hand, releases acetylcholine at their target. Which was similar, not even similar, it's exactly what we saw in the somatic. The big difference between this and the somatic is what kind of receptors are on the target muscle. So the target, it has a subtype of acetylcholine receptors called muscarinic cholinergic receptors. We said that nicotinic cholinergic receptors are always excitatory. These can be excitatory or inhibitory. So when you release acetylcholine at some target for the parasympathetic, it really depends on the particular target. Sometimes you'll actually stimulate the target, like you're going to turn on your salivary glands, and parasympathetic, that rest and digest, is going to promote digestive processes, but it inhibits other things. It's going to route the blood one place but keep the blood from going off to your muscles and stuff. So what happens when you release acetylcholine at your target? It can be excitatory or inhibitory if you're talking about parasympathetic, as opposed to the somatic motor. Even though it released acetylcholine at the target muscles, it always caused the target muscle to contract. So does that make sense? So that's this difference. Even though this releases acetylcholine like somatic motor, it's always excitatory for somatic, but in autonomic, you don't know what is going to be unless you look at the specific situation you're looking at. It's sympathetic, excited. So it's also, these are also depends. Sympathetic is going to excite certain things, and is going to inhibit other things. Like sympathetic inhibits digestive processes, but it speeds up your heart. So these adrenergic receptors are also what we call indirectly gated, meaning that what happens when they bind their little neurotransmitter is going to depend on the specific situation. Wait, what is that? Nikki's saying excitatory. What does that mean?
Haley had asked if it was going to be, is the sympathetic excitatory or inhibitory? And I was just answering her.
So it's not, so sympathetic can be inhibitory, right? It inhibits digestive processes. If you have sympathetic activation, it dries up all your salivary glands. You get all that dry, like if you're really nervous, your mouth gets really dry because you're
Is it
actually, so whether you inhibit or, yeah, so I would say autonomic as a whole can be excitatory or inhibitory depending on what particular function you're looking at, right? So parasympathetic is going to slow down the heart but speed up, you know, but parasympathetic slows down the heart but speeds up digestion, right? So it really depends on what particular function you're looking at. So it doesn't really make sense to think about them being purely one or the other. They both do both. And, it depends on what particular function you're looking at, whether or not one or the other is excitatory. The difference between norepinephrine and epinephrine, in fact, let me get the new page here. NE is a neurotransmitter, meaning you've got some autonomic motor neuron, it releases NE, and then you have some target here, target muscle. It's going to be smooth muscle. And there's going to be receptors here. But in general, norepinephrine is directly released at the target. So it's kind of a one-to-one kind of thing. When we think about epinephrine, this is a hormone. This is going to be released into the bloodstream. By the adrenal medulla. We're going to talk about this in just a few moments. So if I have some autonomic motor neuron talking, let's say this is the heart talking directly to the heart, it's going to speed up the heart. If I dump epinephrine into my bloodstream, everything that has the adrenergic receptors is going to respond. So the heart will speed up, but so will your bronchial passages dilate, and also your pupils are going to dilate, and it's going to, everything that has the appropriate receptors is going to have a response because you have dumped the signaling molecule into the bloodstream. So that's kind of the definition of a hormone, is when you basically put the signaling molecule into the bloodstream, so it just gets broadcast everywhere, and then anything that has the appropriate receptor for that signaling molecule is going to respond. Norepinephrine, a neurotransmitter, we're talking more of about a direct little thing here. All righty. So to finish our, to be clear, the ganglion that's in between the two neurons is always, so in the ganglion, it's always just kind of a relay. That's always excitatory, but that's not what's so interesting. What's interesting is the connection between the second neuron and the target, and that can be excitatory or inhibitory. I should mention, while we're here, before we get into some of the details of sympathetic, let's say, what kind of muscle is going to be the target for autonomic motor neurons?
Smooth or cardiac?
Yes, smooth. So let's put some smooth muscle here. And do you all remember what kind of junctions are connecting the different smooth muscles?
Gap junctions.
Gap.
We got the little gap junctions here. So rather than having one motor neuron talk to one cell like we have in the somatic, what you have in autonomic motor neurons, autonomic motor neuron coming in, we have what are called, these are called varicosities, which are these little swellings. And they basically puff the neurotransmitter all over the place. So we call this a diffuse synapse. So this is supposed to be the neurotransmitter. Right, so you're basically just you're exciting all of these. These all have receptors, plus they're all co-exciting each other. So in smooth muscle getting operated by an autonomic motor neuron, you're usually just exciting a whole bunch of them all at once to all kind of squeeze together. All right, so the last thing we're going to talk about is the kind of connection of sympathetic to the rest of the body. For parasympathetic, we're kind of done. Parasympathetic, there's just going to be, like if here's my target, there's going to be one neuron that starts here, goes almost all there. There's going to be a second little one right there. The sympathetic, on the other hand, there's 4 different possibilities for things that leave the spinal cord to become sympathetic. So sympathetic, I'll try to do this clearly. So here's the brain and the spinal cord. This is supposed to be the vertebrae. So you leave the spinal cord, there's a ganglion, and then you go off. So remember those spinal nerves that were coming out at each of the different levels? There is one possibility is the first neuron can synapse in ganglion at same level and enter a spinal nerve. So you can you go synapse at a ganglia that's at the same level. Let's say you left at, I don't know, at level T4. You synapse at a ganglion that's also at level T4, and then you hop onto a spinal nerve and become part of the cable that's leaving at level T4. So that is one possibility. So the problem with this is it means that you would only be able to, because they only leave at the thoracic and lumbar regions. If this was the only way you could do it, you could only have sympathetic fibers along the thoracic and lumbar spinal nerves. But you actually want to have sympathetic fibers coming out on the cervical areas, down on the sacral areas. So what happens is you have what's called the sympathetic chain ganglia. So you have all of these ganglia all kind of going up and down from the sacral region all the way up to the cervical region. So this is going to be called my sympathetic chain ganglia. And so another option here is, maybe I'll do it in a different color. You can enter the chain, go up or down, and synapse at a different level. And enter nerve. So I could leave here, but then synapse down here and then go on a spinal nerve down there. Or leave here, go up, synapse up here and go onto a cervical nerve. So it's possible that so this sympathetic chain, it looks like a bunch of little bulges. It's also kind of called the sympathetic trunk. I will show you pictures of this and lots of other things in a little bit after our break is done. There are two other possibilities. 3 can pass through the chain and synapse in what's called the collateral ganglion. So you can kind of go and just move through, go into some other thing and continue on. This is going to be called the splanchnic nerve. The splanchnic nerves are not spinal nerves. So these things here are supposed to be spinal nerves. In this case here, in case 1 and case 2, you get into the chain and then you hop on a spinal nerve to continue your path to your destination. In option 3, you're going off into these other ganglia and then kind of meandering through the body to get to your destination. It will make more sense when I show you a prettier picture in a few moments. And then 4 is the one I've already kind of talked about. Can trigger release of epinephrine by the adrenal medulla. Basic idea here is, here's your kidney. The adrenal gland, they talk about, it's like a little hat on the kidney. So you can have the first neuron goes to here. The second neuron isn't really a neuron much at all. It just kind of releases epinephrine into the bloodstream. So that is the fourth thing. Is rather than going to a particular target, is you just go and activate the adrenal gland, the inside part, the medulla. We'll talk about the adrenal gland in more detail when we get to the endocrine system. But you're basically going to activate that to release adrenaline into the bloodstream. So we've covered a bunch of stuff here. Are there any questions?
So for that last one, does it only happen with the adrenal gland, or is it in general it leaves the spinal cord and goes directly to a gland and then it releases the hormone?
No, this is specifically, so there is, this thing that I'm talking about now, I mean, there are things where the sympathetic nervous system can trigger glands or inhibit glands for that matter. But this is about a signaling pathway. This is instead of sending a axon to a particular target to release norepinephrine at the target, instead you can take the first neuron and go to the adrenal gland and then tell the adrenal gland release epinephrine everywhere and just operate everything all at once. So number 4 is just that trigger the adrenal gland to release epinephrine into the bloodstream. So if we go back, number 1 was you just leave the spinal cord synapse at a particular level, continue on the spinal nerve at that same level. Number 2 is you go into the chain, but then you go up or down, and then you go onto a spinal nerve that's at a different level. 3 is you don't even synapse in the chain at all. You become one of these splanchnic nerves that's just meandering through the body rather than being one of these spinal nerves that we talked about. And then 4 was triggering the release of epinephrine by the adrenal gland. Yeah, so the chain is outside the spinal cord. In fact, right before we do our break, I'll show you just a couple of pictures, and then we'll look at these pictures in more detail later. But, this would be the sympathetic chain here. Like here's the spinal cord is right here, going through the tunnel through the vertebrae. And here on the side of the spine are these sympathetic chain ganglia, which are all connected to each other, and you have things going up or down through them. So again, we're going to look at this picture and other pictures, and we're actually going to talk about the gray and white ramus communicans once we come back from our break and finish up our discussion of this. Any other questions? All right, so let's return at 20 minutes to 3. That'll give us 40 minutes. I'm going to stop the recording because obviously it doesn't make sense to record while we're on break. But if I don't turn it on, if someone could kind of like go, make sure you turn it on, that'd be great. So I don't accidentally not turn it back on. I would appreciate that. Otherwise, I am going to see you all at 10 minutes before 3, and we will continue. We'll finish up the autonomic nervous system and then introduce some lab stuff, as well as maybe do some of the sensory stuff. All right, here we go. On pause. All right, so yeah, thank you very much, Jamba there. All right, so what I'm going to be doing is just going over some of the resources that you're going to have, given especially that we don't have lab and we don't have in-person stuff. So one is this Mastering A&P, which is given to us, fortunately for free by Pearson, who's the publisher of Marib. And this study area, I think, is one of the places that you're going to find a lot of use. When you launch, so you can find it on the side, and then when you launch the study area, there's a whole bunch of different stuff in here. You can study by chapter. There's art labeling activities, practice tests and quizzes, animations and videos, One of the important things to remember is nothing you do in here comes back to me. So while you will be getting quizzes and stuff that I'll be assigning you through Pearson Mastering, if you go and take practice tests and quizzes or do art labeling activities, it's just for yourself. It's just a way for you to get familiar with the material. So as long as you're playing around in the study area, it's purely for yourself. And they're pretty cool. In fact, some of the stuff, that CNS quiz that I sent out for you guys is like bits and pieces that I grabbed from activities in here. I'm still getting used to what exactly is in here. Obviously, I have to go through in real time and try to look at each and everything and seeing what seems more or less relevant. But a lot of it is very relevant to what we're doing. The other thing that's really nice in lab tools, this is where you have practice anatomy. So this practice anatomy lab, this is this thing that actually our very own Jet Chin helped create. And it uses real cadavers, and it has pictures, and you can make flashcards. Like if we go into practice anatomy, PAL 3.1 here, la la la. We can go into the nervous system, go into the, autonomic nervous system. Here's the brain, and wait, I don't know why this is in the autonomic. So I could actually say, so what is this thing right here that I'm kind of putting my cursor up and down on? Does anybody know?
Olfactory tract.
That's the olfactory tract, exactly. What about this thing here, this kind of X-like thing of this big fat band?
Optic canal.
Oh, the optic chiasma.
The optic
chiasma. Right. So, this is a place where you're going to be able to, even if it's just pictures, at least kind of remember what real stuff used to look like. What part of the brain? Oh, I didn't even. But did I do that? Did somebody do that? No, I'm doing that. So again, fortunately for you guys, you don't have to recognize all the different cranial nerves in this picture. The only ones you need to know is the optic chiasma and the olfactory tract. But let's see what else is here. Oh, actually, what do we call this big flat sheet here in between the two hemispheres?
Falx cerebri.
The falx cerebri, exactly. What do we call this kind of membrane? It goes in between the two lateral ventricles.
The septum lucidum.
The septum pellucidum.
One question, is that septum pellucidum, you can also call it as third ventricle?
No. In fact, it's not even near that. The third ventricle is going to be in the diencephalon, right? It's between the two walls of the third ventricle are the right and left thalamus. So this thing that's kind of glowing blue here, that would be where the third ventricle is.
Oh, okay. I was
Right.
thinking something else.
So
Okay.
this is the lateral ventricle. If you dug through this little curtain, you'd be in the lateral ventricle.
Okay.
And then, here you can go down. Here's that aqueduct of Sylvius down into the fourth ventricle and the arbor vitae. So
Yeah, that's what I meant to say. I'm sorry to interrupting. You can call third ventricle septum pellucidum.
no.
Interchange? No. Okay.
The septum, maybe let me.
Because I was watching this video on YouTube and somebody was like mentioning it as the third ventricle as septum pellucidum. You can call it interchangeably.
That person was incorrect.
Incorrect.
Anybody can say whatever they want when they're on a YouTube channel.
Okay.
So that just so it's not what
Thanks
it
for
is.
correcting.
And I'm trying to nab it. Here we go. I'm going to I will show you the septum pellucidum since you should know where it is. If we go into remote instruction, anatomy, Pearson, Marine PowerPoint, and nervous CNS. So, and then, come on, little puppy. Here we go. So ultimately, it's going to be the thing that kind of separates here between the right and the left lateral ventricles as the cerebrum forms. So doobie doobie doo. So it's like right here. It's right in between the right and left lateral ventricles where I'm kind of drawing. That would be where the septum pellucidum is. If you dug through it, you'd be in the lateral ventricles. The third ventricle is down here. The third ventricle is where you have the right and left thalamus on either side. So here again, here's the third ventricle in the diencephalon. The septum pellucidum is actually in between
The lateral ventricle. Okay,
the lateral
thank you for
ventricles.
correcting.
Does that make it clearer?
Yes.
Yeah, in fact, here you can see the septum pellucidum as this little like curtain in between the right and left lateral ventricles. So, all right. So back to where we were, which was where? Okay, so actually, can whoever is, if someone could mute, somebody is, cool, thank you. So this isn't perfect. In fact, I forget who it was. Somebody asked me to look into like complete anatomy. So I actually reached out to them and I got like a thing to explore it for a little while. And it's so much nicer than this. But it's expensive and they don't offer it for free to you guys. So I'm looking into seeing if there's some way maybe the school will cover it or something. So there are other tools that are prettier than this and that also do this other program does a really nice job of going back and forth between the models and the pictures and the actual cadaver specimens. This is, like this kind of reminds me of, like, it's cool. Like if you played Donkey Kong back in the 80s, Donkey Kong's awesome. But then when you actually realize like what is possible now with high resolution, like video games, you realize, oh, it was actually pretty crude. Like these things are pretty cool, but they're also kind of crude compared to some of the other stuff that's out there when I started looking. But that being said, it's pretty good. So this is showing like sympathetic chain, like right here, sympathetic trunk and ganglia. Again, sympathetic trunk and sympathetic chain are just synonyms for the same thing. Again, and like I said, from this picture, it's kind of hard to really orient yourself. That's part of the reason I don't like doing it this way. If you're looking at a three-dimensional thing, you would be able to see it much more clearly. This is just kind of showing the adrenal glands. But in general, this is going to, let's see. So Sakai, that was you. So in general, it's going to be worth going into this and getting a feel for different things. I will assign you things. There's one thing, again, that I've assigned. I created a quiz I called CNS quiz. You should do that. Over the next few days, I'm going to be more intentional. That was just kind of me like trying to see how does this work. I'm going to be more intentional about creating kind of targeted quizzes out of this system that will actually test you on the stuff we've been covering, just to give you something you can do that will help you study, make sure that you know the stuff that you need to know. So I have not figured out, supposedly you can integrate this and get it directly from Canvas, but whenever I've tried that, it's not working right. So I'm going to continue trying to figure that out and talk to our IT people and see if we can just launch the Pearson stuff from directly within Canvas. But for now, at least, you have to launch it from its own portal. Other thing that I created is, where is it? Why, look, there is a human anatomy channel on YouTube. So I've made this so it's like where the edited lectures are going to go. This is like the half a lecture from Tuesday, and I'll put today's lecture up here as well. So I have two playlists. I have one playlist that's just anatomy lecture videos, and then I'm going to start accumulating videos that are going to be useful for lab and stick them and add them in this playlist here. So far, the only thing here is this thing that Tina Christensen, who's one of the other anatomy instructors, she made a little 7-minute or actually 11-minute thing that kind of just guides you through a lot of the stuff in the Pearson mastering that she thinks is useful. So it's probably worth just going and checking this out and getting her point of view of this, so you can see different things that are available to you. But you probably do want to sign, actually, I'm not exactly sure, but subscribe. I kind of feel like I'm one of these like YouTube guys. Make sure you subscribe to my YouTube channel. But I think this is going to be a useful place to come, as over the next month, more and more stuff's going to show up here, both the recordings as well as a variety of other material that will be useful. Recordings are also available if you go into, if you're in anatomy and you go to ComforZoom, and you go into event recordings. There are also, these are kind of the raw recordings of whatever we're doing. So that's always here. But what I've been trying to do is kind of clean them up a bit and then upload them onto YouTube as a better repository. But this is another place you can come. If something's not on YouTube yet, you can come to ComforZoom event recordings. And the recordings are only for when I've hit the button. So unfortunately, we've missed half of the first lecture. So it looks like Jamba's head is like kind of exploding. So all this smoke coming at the top. That's funny. So other things. I put the file, in fact, let me go to student view, courses, home. If we go to student view, I have made sure that the files, select the files thing is still here. If you click on files, there's a variety of things that are going to be useful for you. One is there's the Martini PowerPoints, which we had before. I am now starting to upload the Marib PowerPoints as well as we go through them. So you'll have another version, like a lot of the images I've been using are from the Marib. Histology iPhone pics. This is something I've just been adding in as we've just been going right now. Over the years, whenever I'm like seeing cool slides, I often take pictures of them, and I've been kind of accumulating them. So, and it's turning out to be a really nice thing now. Like for instance, we've been talking about ganglia. It's like, oh my God, there's a ganglion. Can I zoom? I don't know if I can. Oh yeah, I can. I'm going to zoom here. Oh, that doesn't work there. Instead of doing this, I'm going to actually go into my actual directory where I have them, because then I can. Hold on one second. Histology. Nervous. So if I go and, some of the stuff we've already seen, like there is, what are we looking at here actually? Does anybody remember this? We saw this once before. This could end up on your final exam. If I had this there and there was like a little pointer right here. What is that? Anybody?
I think Nina said it, the node of Ranvier.
Yeah, exactly. A node of Ranvier. So these, and what is staining black in all of these?
That's the myelin sheath, right?
Yep, those are the myelin sheaths. So this was that teased nerve. So that's, so I have that in here. What's another? Let me get back to mouse. Wait, mouse. There we go. What? Hold on, wait, this is not correct. Hold on. Sorry about this. Okay, so, or if I brought this in, what are we looking at here? Does anybody remember? This is easier if you zoom in. You start noticing each of these little things is actually kind of like a little donut.
I don't know if we're seeing the same picture that you're talking about on the screen.
I think we're
you're
still
not.
on node.
Thank you very much. So what are we looking at here?
Is that the white matter of the spinal cord?
So you're very, very, very close. So it is totally myelinated axons. And if I zoom out a bit, you'll see that it's not quite that. So let me zoom out a little bit. So you can see we're not in the spinal cord, and this whole thing is a bunch of those little myelinated axons, but wrapped up in a big thing of connective tissue. So what are we actually looking at, do you think? What would be a whole lot of myelinated axons all bundled together and then wrapped in a dense connective tissue outer coating?
Nerve?
Say it again, Casey.
A nerve?
It's a nerve. This is totally a nerve. So this is, when we've been talking about all those spinal nerves or radial nerves or any of this and that a nerve, this is that nerve. So we're kind of bringing back the stuff when we first started looking at nervous tissue. Now we're looking at it in more of a context. So this is the nerve with, what do we call the outer coating of the nerve?
Epineurum.
Yeah, it's the epineurium. That's the epineurium. This stuff in between would be the perineurium, and around any one individual of these things would be the endoneurium. So that's we got that going for us. Um, I should have a better picture of this. This is the spinal cord of a young cat. You know, again, if you look at this, we can see stuff. In fact, part of what you're going to need to do during your kind of lab time is look at a spinal cord like this and be able to orient yourself. You know, what part is this?
Ventral horn.
That's the ventral horn. And I know for two big reasons. One is it's more rounded and the other side is more peaky. I also can see here very dramatically that the anterior fissure is much deeper. You've got this real crazy, you don't always see that. You know, if the thing is really squished in on itself, even though there is a fissure, it might not look like it because they're pushed on each other. What kind of cells are lining the central canal here?
Ependymal cells?
The ependymal cells. And again, this is all the white matter, the columns. So now you have more words. We've got like, you know, columns, and we have dorsal horns and ventral horns. You know, so if we were in our real lab, we would go and look at spinal cord again. And we would be able to get a sense of, oh, not only do we see gray matter and white matter, but now we know the different parts, and we know this is motor and this is sensory. Next one, what is sitting right next? What do you think this, let me erase my drawings. So here's the spinal cord. What the heck do you think this is? And it's in the right place. It's sitting where it's supposed to be. It should be sitting right next to the spinal cord.
Part of the sympathetic chain.
Right, so it is a ganglion. So this one, it turns out, is not. The sympathetic chain would be a little further away. Like, in fact, if I, let me share whiteboard. So like if this is spinal cord, and it's sitting in, this is supposed to be a vertebra. Obviously there are little gaps where the nerves can act. Sorry about that. I'm trying to leave like a little gap. So we have this spinal cord here. We have the ventral roots, the dorsal roots. Do you remember the dorsal roots? What kind of nerves make up the sensory neurons that make up the dorsal roots? Anybody remember?
Unipolar.
Yeah, they're the unipolar. So we have right around here, we're going to have the dorsal root ganglion. And then, the roots come together to become the spinal nerve. So the sympathetic chain is going to be more, it's kind of along the side here. So the sympathetic ganglia, they're on the outside of the vertebra, whereas the dorsal root ganglion is actually really close. It looks like a little bulge right coming right off of the root. In fact, let me go back into the I need to file open nervous PNS. Does it have it here? No. Oh, so it's probably in the CNS one. Let me look in the CNS one. Sorry about that. File. Doobie-doo. Yeah, so right here. Here, in fact, we can go say slideshow from the current slide. If this is the spinal cord right here,
We can't see the screen you're talking about.
this is the spinal cord. Here are the roots. And like right here, dorsal root, these are, and then coming off.
We're still stuck
right
on your
near,
whiteboard.
oh, fuck. Excuse me. Thank you. Oh my God. All right. Here we go. So I'm going to erase everything I just did because I just totally spazzed on you all and you have no idea what I was just talking about. All right. Erase all ink on the slide. All right, so here's the spinal cord coming down. Here's the dorsal root. These are the axons that are coming into the dorsal horn. And if you look, this bulge here, that is the dorsal root ganglion. So the dorsal root ganglion is, it's still outside of the spinal cord, but it's really close to the spinal cord. It's actually still in the canal within your vertebrae. So on a slide, on a microscope slide of the spinal cord, sometimes you actually catch the spinal cord as well as the dorsal root ganglion as well. The sympathetic ganglia are going to be outside of the vertebrae, so it wouldn't show up. You'd have to see it on the other side of the bone. So now if we go back and if I go share this picture here, here is, here's the spinal cord. This then is what? What does it have to be?
Sympathetic ganglion.
No, it's
Dorsal
not this.
ganglion.
That's the dorsal.
It's the dorsal root ganglion, exactly. So let me annotate. Let me go back. I'm gonna annotate. So this is the dorsal root ganglion. And if I zoom in on it, what you can see is each of these things, these big round things are actually neurons, unipolar neurons. I'm going to show you another picture of a dorsal root ganglion.
Does the sympathetic look similar to that?
Yeah, in fact, I'm going to show you both of the sympathetic and the dorsal root ganglion in just 1 second here. hold on, I gotta find my, here we go. Okay, boop, ba-doop, ba-doop, boop, 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 boop. Wait. Oh, I see. I know what's going on. Here they are. Okay, I'm going to go and share screen. Boom. All right, so this is one of my favorite slides showing a dorsal root ganglion. So this thing right here. Wait, and that's, I wanted the photo. There, that's, well, I'm gonna share something slightly different because this is, I can't, yeah, this one. So this I can zoom in on. So each of these big round things is a what, do you think?
Those are the neurons.
Yep, that's the cell body of the neuron. That's what that is. And so each of these big round things is a neuron. And then what do you think all those tiny little dots that look like little moons, like little satellites in orbit around all the neurons?
Those are the satellite cells.
Yep, those are the satellite cells. And then here you can just, what do you think this is over here?
Can't see what you're pointing at.
I'm going to clear and I'm going to draw like in here. Oh, you can't see that. Duh.
Nope.
Hold on. So right in here. Anybody have an idea? And here was like the cell bodies. What do you think has to be coming in and out of the cell bodies? That would look like a bunch of long
Axons.
yeah.
Axon.
These are all the axons. These are the axons that are coming in and out to create that dorsal root. All right, so this is a really nice, this is one of the most beautiful pictures we have of a ganglion. This is a dorsal root ganglion showing the nerve cells are the big round cells. The satellite cells are the little dots around, and then the axons that are coming in and out. If we, oh shoot, excuse me, I'm still figuring this all out. I am going to share. You know, this thing here, this is stained with silver, but this is also a ganglion. This is a sympathetic ganglion, but you can still see the basic idea is it's got the kind of big round cells and also axons around. So sympathetic ganglia or dorsal root ganglia look kind of similar. They're big cells surrounded by satellite cells. So what you do want to do, like if I was going to give you a ganglion on an exam, I would probably give you one more that looks like that. Whoop, no. Hey. I I'm having, I don't know why this is becoming a pain in my butt here. Let me, yeah, it would be something more that looked like this, where it's very obvious. You've got these big cells surrounded by the little satellite cells. So this would be like a classic looking ganglion. Um, so what else? The other thing, what time is 3:30? The other thing we can talk about before we leave this spinal cord and ganglia and nerves, there's one cool little thing that happens for the sympathetic motor fibers to go, because like if, so in fact, let me do this right now. We're gonna add one more little piece of the story for share content.
Is that dorsal ganglion in the?
This is a dorsal root. Yeah, that was a dorsal root ganglion. Exactly. That thing you just saw was a dorsal root ganglion. So now, again, here's the spinal cord. We've got dorsal root ganglion. We have our motor ones coming, the ventral roots here to make the spinal nerve. Here's my spinal nerve. This here is my somatic motor. Remember, somatic motor is just one shot. You have the cell body in the spinal cord, and then this thing goes all the way to the muscle that is its target. We talked about it at the beginning of today. Somatic motor starts in the CNS and goes all the way. I kind of refer to the idea of the lateral horns. That's where the sympathetic motor fibers start. So you've got this guy coming out. So can it just get onto the spinal nerve and go to its destination? What does it have to do first?
Needs to synapse with the second neuron.
Yeah, exactly. It's got to go and synapse in the ganglion. So what happens is this thing takes an off ramp and it synapses in the sympathetic ganglion. This is the sympathetic. Again, I usually call it sympathetic chain ganglion, or it could be the sympathetic trunk. And then it gets back onto the spinal nerve to continue on its way. So if it's a sympathetic motor fiber, it can't just go onto the spinal nerve to get to its destination. It has to take an off ramp, synapse in the sympathetic trunk or sympathetic chain, whichever you want to call it, and then take an on ramp back onto the spinal nerve. And so there's names. There's names for everything. So I'm going to give you the names for the off ramp and for the on ramp. So the off ramp are myelinated fibers. So what color do we usually, if we have white myelinated, oh, I just said it. If we have myelinated axons, they're usually white. So this is going to be what we call the white ramus communicans, or white ramus communicantes. So white ramus, hey, not ramps, ramus communicans. Ah, there. So white ramus communicans is the off ramp.
And that's the first neuron, right?
That's the first neuron, exactly. The first neuron is going, takes the off ramp, takes his white ramus communicans, it's not cons, it's cons. Dagnabbit. And then this on-ramp, the on-ramp back onto the spinal nerve. It is non-myelinated. So what do you think its name is going to be?
Gray matter.
The gray ramus communicans. So that is that. So are there any questions? At this point, we're pretty much done with all of the motor stuff. Let me show you some more pictures, maybe. Or actually just a picture you've already seen, but is going to be, the more we keep talking about this stuff, the more, like again, this idea, the sympathetic trunk or sympathetic chain, these are hanging out right next to the spinal cord. So you have right here, this thing that you see here, this is supposed to be the white and gray ramus communicans. So let me, pointer options, pen. This one that is white, this is supposed to be, you know, you've got motor things coming out, going into here, synapsing, and then the second neuron coming on the gray one and then continuing down the spinal nerve. So does that make sense there? Again, each time, in fact, like gray ramus and white ramus communicans. The white ramus is just the sympathetic motor for the first neuron coming, synapsing in the ganglion. The gray ramus is the second neuron heading back to get onto the spinal nerve to continue on to its destination. So, make sense? Yes. Okay.
So it just turns around in the trunk, the synaptic trunk.
It makes like a loop. Yeah, for whatever reason, it kind of does this. It kind of comes. So it's like, this is the white ramus is actually over here. The gray ramus is coming back. So I know that's kind of confusing, right? It basically comes out, synapses, and then comes back and continues on.
And you also said something about it only occurs on thoracic and the lumbar region,
Um,
lateral.
so yeah, so although not
Correct me if
it's
I'm wrong.
that's not actually going to be true. It's the first neuron originates at the thoracic or lumbar region, but if you remember, like option 2 was you can enter the chain and move up or down. So you could enter the chain and move up someplace, and then go synapse at a different level of the chain and go on. So yeah, so you actually can have things coming in and out at different levels. So yeah, so you can, even though the original motor neuron has to have its origin ultimately in the thoracic and lumbar region, because it's like this elevator system to go up or down, they can enter into spinal nerves at any level.
The
So-
parasympathetic neurons can't do that, right?
Correct. The parasympathetic neurons are just heading all the way to where they're going. Like if I go, this card, let me go into the PowerPoint autonomic slideshow. Right, there's no sympathetic trunk or anything. They just go zip, meander through the body all the way to their target and have their second neuron right near the target. So it's a very different scenario. Whereas the sympathetic, they come out, they have the sympathetic chain right next to the spine, like all of sympathetic trunk ganglia. It's all of this stuff going up and down. Maybe I should make it a different color. All of this is all the sympathetic trunk or sympathetic chain. Whereas the parasympathetic is just going on its way. Yeah, so here this is kind of showing, comes in, but can come out all these different levels. This card. Let's see. So where we are at 344. Any questions about motor? In terms of lab stuff for this, you should be able to look at a picture of the nervous system and recognize the sympathetic chain. Again, it's going to be those little like clusters of neurons that are connected to each other, that go all the way up to the cervical, all the way down to the sacral, and are all interconnected with each other. You should, under the microscope, you should be able to recognize a ganglion. I'm still trying to figure out, after we're done with today, I'm going to go back into that mastering anatomy and physiology and create quizzes and stuff. So by this weekend, you will have content. I'll send you an announcement once, all this stuff is prepared. but like I do know that lab is still, we're still trying to figure out exactly what lab looks like. And as of right now, normally I wouldn't keep talking and talking. I'd be having you like looking at stuff or looking at, even for the spinal cord. We actually have some really nice models of the spinal cord where you can look at it and see the different parts. And then we have really nice real spinal cords. So you'd get to spend time looking at, oh, I see the actual roots, and I see the actual fissure, and I see the actual phylum Terminale and Cauda equina and Dura mater. So I'm still trying to figure out the best way to get you at least have some sense of that. I'm going to spend some time this weekend also looking at videos like YouTube stuff that's out there that might have little guided tours, with cadavers that you can film. Obviously, with our cadavers, even if I went into lab, I'm going to get fired if I post pictures of our human material. So I need to find stuff that is actually okay to post online here. So
I had a question. Do
Uh-huh.
you mind going over the splanchnic nerve and how that plays into everything again?
yeah, so I will show that right now. So slideshow from current slide and share screen. So like right here, if I, actually let me use a different pointer. Let's use let's use like red. So there are these three different options. One is you enter into here, and then you end up on a spinal nerve. But sometimes you go through without synapsing. And then you synapse in a ganglion that's outside of the sympathetic trunk. This is an example of what we'd call a collateral ganglion. So pointer, pen. So the celiac ganglion is just one of them. There's the inferior mesenteric ganglia. There's a variety of these different ganglia that are outside of the trunk. So this is sympathetic, but not the sympathetic chain. And so you have this neuron that goes through without synapsing in the trunk and synapses in one of these collateral ganglion. And then the second neuron just kind of meanders through the body to its target. It's not on a formal spinal nerve. So that's what the splanchnic nerves are, the ones that are kind of meandering and are not these kind of formal spinal nerves that are coming off of the different levels of the spinal cord. And what if I can maybe show some pictures of the collateral ganglia. Like here, this celiac, like right here, this is a sympathetic ganglia. Here's some more. These are all sympathetic ganglia. They're still kind of near the spine, but they are not the sympathetic chain. And so the stuff that is the sympathetic chain The stuff that synapses in there is going to end up on a spinal nerve. The stuff that goes and synapses in a collateral ganglion just becomes one of these meandering splanchnic nerves. That's all it is. It's a meandering nerve through the body rather than one of the formal spinal nerves. So so that is that. What else? Maybe, what time is it? Maybe the last thing we'll do is I will finish the eye, just since or I don't know. I mean, I should ask, where are people at?
Do you mind if you revise the sympathetic, how they turn back the nerves? I don't get it though.
Wait, so say that one more time, Elsa.
The sympathetic,
Uh-huh.
how they turn the nerves, the last one we explained before that.
I'm not sure I understand your question. So say that one more time.
Earlier you explained about the sympathetic, how the nerves, they turn back round or something like that.
I mean, I was talking about how the first neuron has to leave the spinal cord and then goes first synapses in the sympathetic trunk. That was the white ramus. And then then the second neuron kind of hopped back onto the spinal nerve was the gray ramus. So maybe that's what you're, like, if we go right here, let me share this picture here. Hold on a sec. Right, you have your first neuron. This is a first neuron number 1, sympathetic motor, comes out, leaves the spinal nerve through the white ramus, synapses in the sympathetic ganglion, sympathetic trunk here. Then the gray ramus gets back on, and then it's on the spinal nerve to continue on its way. Is that what you were asking about?
Yes, thank you.
Okay.
Is there ever a time where it just doesn't get back on?
So the splanchnic nerves are where they, instead of doing that, they just go through and then they synapse someplace else.
Okay, thank you.
Yeah, so not all of the sympathetic fibers travel on spinal nerves. Some of them are on those other nerves. Okay, so let's see. So let me check in with you all. Like, again, normally I know that, we talk, but then there's time to actually just sit and do stuff. So I don't know if everybody is getting like pretty fried. We could just call it here and then on Tuesday kind of finish up the senses, especially since we've covered a lot of the eye already. Or I can kind of introduce the senses now and then have that be kind of the end of today. So kind of get a sense of how people are holding up. How are people doing out there?
I think it could be good to have all the senses kind of together. So maybe starting that next class and then have us review what we've done this class, maybe by going on Pearson or using our own like lab book that we have to just kind of go over everything we've done this class. Okay, so what I would say, what you're going to want to do is definitely, think about histology.
I have those slides. Oh, there's one other um, slide option for you that is actually really good. So in fact, I'm going to share this with you. Where is it? There it is. I need to go into here. And I got to go into here. And I need to go into here. Okay. I'm going to share our. So this is, if you go into modules and you go down to oh, do I not have it here yet? Okay, hold on. Give me 2 seconds here. So I need to leave student view here. I'm going into, you get to see how the sausage is made here. Histology references, I'm publishing it. So now it is going to be visible in student view. And now we're going to go into student view to make sure you can see it. Modules. Down here under histology resources, when I was just Googling this morning, I came across this thing from the University of Minnesota that's actually good. A lot of the stuff, there's a lot of people who've put histology on the web, and a lot of it just really, really sucks. And the amount that I've been looking through this website, I've actually really liked it. The person who put this together actually cares about whether or not the slides are good quality. And so if you go in here, like in the slide box, nervous tissue, there are spinal cords and spinal cords with different kinds of stains. There's dorsal root ganglion. Ultimately, there's lots of other things as well. But for today, in fact, one of the things that's kind of fun, this one has the violet stain. I don't know if you remember back when we first introduced neurons, I said that when they first started staining the neurons that you'd get this kind of dark staining stuff that they would call Nissl substance or Nissl bodies. And it turns out, let's see how we do this here. We zoom in and in. I don't know if, yeah, you can see the kind of like dark staining little dots inside the neurons. That is what the early anatomists called Nissl substance or Nissl bodies. We now know that it's just, rough endoplasmic reticulum. It's the RNA actually that's staining, and it's because these things are making a lot of proteins and stuff. But it's kind of fun that you guys actually get to see actual Nissl bodies stained, on this. So, here's the central canal with the little, ependymal cells here. And, so I recommend, it doesn't have everything we want, but the stuff that it does have is actually pretty good. So I would say click on that link, which is now available to you in the histology module, and I'll be putting more stuff there as well. And I'm going to be trying my best to, as we keep going, to organize things so you don't have to like find things all over the place. Right now, that's not the case. Right now, things are still pretty random and chaotic. But this weekend, especially, we've survived this first week, and I've got a better feel for what things are. I'm hoping this weekend to get a lot more stuff feeling like organized and kind of resources to kind of give you more of a roadmap to how to make sure you're getting the lab material, even though that's been kind of the trickiest thing for me to think about. Yeah, so I would say make sure you don't forget the histology. Again, I will be making, so let me go back to my whiteboard.
Quiz section on that site looks pretty good too.
Say that one more time.
I said the quiz section on that site looks pretty good too.
Yeah, so that, yeah, I mean, and the problem with quizzes is every teacher has their own kind of, kind of quirky thing of what they think is really important, what other people think is important. Like that's one of the tricky things for me, just kind of giving you pre-made quizzes, because there's some people that, have quizzes on things that I think are kind of like ridiculous, like who needs to memorize that, and leave out stuff I think is really cool. So, but yeah, make sure you're always, make sure you use, the study guide more than ever when you're trying to figure out what are you responsible for. And as we start going into all these other resources that I didn't create but still are, finding useful for you, make sure you don't put a whole lot of energy memorizing a bunch of nerves that you don't need to know. Make sure you like look at your nerves to know, and make sure you just spend the time learning the ones you need to know. So be careful about like accidentally like filling up your brain with a bunch of stuff that's going to take up the space of other stuff. Make sure you get your Pearson account. At this point, I think most of you have it, but by tomorrow, I will be have created some quizzes that will help you review the material we've been looking at this week. So in addition to this being a great study guide, like with that, kind of study area that we talked about, it is also going to be a necessary part of things that you need to do and turn in. In terms of the points, I have not, don't worry about the points or what the points mean. I have not figured out how the points that accumulate on the Pearson website are actually related to points that are part of your overall grade. I will also be trying to figure that out over this coming weekend. So for right now, don't like think about like points meaning anything. But anyway, are there any other questions before we like say wish you adieu for the weekend?
A Pearson assignment is due tomorrow, yeah?
Say that one more time.
Is that a Pearson assignment due tomorrow?
You should, yeah, if you haven't done it yet, you should do it, the CNS one, just so you get a sense of how it works even. So here's the sun setting over the hills. Oh, it's turning red. Looks like the sunset for first week of online instruction. Little birds flying in the air, but nobody out there because they're all at home sheltering in place. So if you have questions about things, please feel free to email me. And again, thank you for your patience as we're figuring this all out. And I will see you all again on Tuesday.
Thank you. Have a good weekend.
You too.
Thank you, Professor. Have a good weekend.
You as well. Bye bye then.